Hello, my name is Joshua Mugabe. I welcome you to Poriferans. What are Poriferans? The phylum Poriferans contain the simplest creatures of the animal kingdom. Sponges, they are aquatic sessile beings, meaning they are not able to move by themselves, and they keep themselves attached to substrates. They do not have tissue diversity, and their bodies have pores. They are multicellular, like all beings of animal kingdom. What is the way of life of sponges? Sponges are exclusively in aquatic environment, and they are attached by their base to a substrate, that is fixation ground. Sponges are filtering animals. They nourish themselves from nutrients that enter their atrium brought within water. What is the typical shape of Poriferans? Sponges have bodies in form of tubular vessels or grubs, open in the upper extremity. They have an internal central cavity and porous walls. The central cavity is called sponge coil, and the openings in the upper extremity is called ossicrum. How does water move inside sponges? What is the function of pores in these animals? Sponges are filtering beings. They make water enter their bodies by their lateral pores. What then circulates inside the central cavity and exits through the ossicura. How do sponges try to protect themselves against harm or from the environment? Is that method efficient or rudimentary? Sponges, they can close their pores to avoid entrance of water into their bodies in the presence of stimulus that may mean danger. This method, however, is rudimentary, but it is actually a protection attempt against nuisance agents. What are the main cells in which Poriferans are made? Sponges have their outer wall covered by flat cells called pinacocytes and having pores well delimited by special cells called porocytes. The internal wall is filled with choanocytes, flagellated cells specialized in phagocytosis of food brought to the central cavity. The choanocytes flagella also maintains the water flux inside the sponge. Between the outer and the inner coverage, the poriferan body, there are cells with amoeboid movements, the amoebocytes. Since they are embedded in connective matrix, amoebocytes move and distribute nutrients to other cells, and they also produce spicules that, like a primitive skeleton, fill the tissue and support the body structure. Concerning digestion, how are poriferans characterized? Sponges are different from other animals since they present intracellular digestion. They don't have a digestive system, nor do they release digestive enzymes in the spongicoil to cause extra breakdown of nutrients. How are animals divided according to their digestive processes? Apart from sponges that do not have a digestive cavity where extracellular digestion takes place, all other animals have digestive system with an internal cavity in which extracellular digestion occurs. How are gases exchanged in sponges? The gas exchange in sponges happens by diffusion from the exterior to the cells that absorb molecular oxygen and liberate carbon dioxide. Do sponges have nervous, circulatory, and excretory systems? Sponges do not have a nervous system, neither circulatory system nor excretory system. Is reproduction in sponges sexual or asexual? Reproduction in sponges can be sexual, that is by budding, gemmation, or fragmentation, that is regeneration, or it can be sexual with larval stage, that is a ciliated amphiblastula larva. What is the evolutionary advantage of the occurrence of sperm cells and larval stage in the life cycle of the sponges? The sexual reproduction in sponge, in addition to contributing to genetic variability, also facilitates the colonization of further environments by this being, since sperm cells and the larva are mobile and can swim in the exterior to compensate the immobility of the adult individual. What is the economic importance of sponges? Some chemical substances are secreted by sponges have anti-inflammatory, antibiotic, anti-tumoral activities, and they are used in the production of medicine. Since ancient time, the endoskeleton of some sponges have had commercial value. They are used as cleaning implements for the bath, that is bath sponges, to wash animals, objects, and so on. Sponge identity card. How are sponges characterized according to examples of representing beings? Basic morphology, type of symmetry, embryonic germs, layers, column, digestive system, respiratory system, circulatory system, excretory system, nervous system, and type of reproduction. Examples of representing being: sponges, they have a basic morphology of being tubular or globular body with sponge coil. They are sessile, clonocytes, pinocytes, and amoebocytes. Types of symmetry not established. Germ layers and corem do not apply, since Poriferans do not have true tissue organization. The digestive system, not in existence. Reproductive system, non-existence. Circulatory system, non-existence. Excretory system, non-existence. Nervous system, non-existence. Type of reproduction, asexual and sexual with larval stage. Thank you very much for participating in Poriferans. It's a question and answers to build your biological knowledge step by step. Please do like, share, and subscribe.